So it's a real pleasure to welcome Dr. Philippa Hulley today to have a short chat about some of her research. Was recently funded through the Paget Association. So Dr. Hulley is an associate professor and university lecturer in musculoskeletal sciences at the University of Oxford. And Philippa, if I may, was recently funded for a project titled Developing a fully human 3D bone organ model of Paget's disease. And over the next few minutes, then, we're going to have a question and answer, a chat about some of Philippa's research. So welcome, Philippa.
Thank you very much. It's really great to be talking about this work with you today.
Thank you, Philippa. As I mentioned, your research was recently funded in part by the Paget Association and your research group in a project that's titled Developing a Fully Human Three-Dimensional Bone Organ Model of Paget's Disease. So a bit of a mouthful there, but really your research relates to growing bones in a dish. So new models of bone diseases and Paget's disease. And ultimately, I guess we'd hope that the models would give us a better understanding of Paget's disease mechanisms. So first of all, could I just invite you to very briefly tell us about these exciting new models that you've been able to develop at the University of Oxford?
Yes, sure. So people have for many years, in fact, decades now, been able to create little bones in a dish, and that in itself is not particularly novel. But human bone cells have been particularly difficult to encourage to form bones and have needed a bit of optimization and tips and tricks. And we wanted to use patient-derived human cells. And we found that if we actually seed the patient's cells onto a jelly and give the cells some plastic hangers to grip onto and hang around, that they'll pull the gel around the plastic posts and contract it into a little thread, which then over a couple of weeks, they convert into living bone. And they're perfectly happy then and remain alive for over a year, we found. So it's quite a workable system.
Wow, that's really interesting. So can you tell us about the cell types that you're able to incorporate into these bone models and how they relate to the cell types that are perhaps most relevant for Paget's disease?
Yes, so first let me just show you how robust these little systems are to handling. We made a video in the lab just showing the constructs hanging on the plastic hangers over wells of pink culture medium. And you can pick them up and you can move them around. They're quite robust. You can combine them with different cell types or treat them in different ways over time. So they're proving to be quite useful in the lab in a variety of experiments.
Wow. They actually look like bones. The shape of them is, we call them mini bones, but actually they seem to resemble bones. Is that because of just how you configure the matrix that they grow on?
Partly, I suppose it's the length and the fact that there's a swollen little bit at the head, but they mineralize and go white and do look and feel and behave very much like a small bone. And you asked about the cell types that we're interested in. Most of the focus in Paget's research has been on bone resorbing cells, the big osteoclasts that eat holes in bone. And in fact, diseases like myeloma, the osteoclasts eat holes in the skull there. You can see holes punched by the bone resorbing cells. This happens to some extent in Paget's, but it's not the whole story. So although the drugs called bisphosphonates are effective in treating both diseases, nothing fixes the deformities that happen in the bone. It's as if the bones have forgotten how to be the right shape to be. And the cell type that's missing in all the models so far have been osteocytes, which are by far the most abundant cells in human bone. But they live deep in the bone in little caves. They look a bit like spider crabs, and they have a huge mechanosensing function. And they only exist buried in the depths of bone. And most culture systems don't allow this to happen, but ours does. So we form these little spider crabs in their caves, in our mini bones, and are therefore able to look at these for the first time in Paget's.
So human osteocytes, as you say, are very difficult to study. Are you able to just very briefly explain what the trick has been? What is it about your model that allows you to incorporate this kind of key component of a human bone model?
We don't understand it fully, but one of the most important things is time. They have to be allowed to bury themselves in their own time in bone and stay there for a while. And within 2 weeks to 4 weeks, all the mature markers of osteocytes start to come up. But if you try and do them on a flat dish or on a surface, or they don't have enough bone around them, or the bone formation is not active, they don't form.
So these are really potentially kind of a powerful model to study bone function and bone dysfunction, and obviously Paget's disease being a condition of a bone disorder. What sort of questions might you be able to ask about Paget's disease using these models that you've been able to develop, and the sort of future view of the questions?
Yes, there are a lot of questions that we can now effectively ask, and that includes looking at these cells in isolation, but also combining them with other cell types. And one of the very first exciting observations we made is when we put a Paget's mutation, a common Paget's mutation in sequestosome 1 into stem cells and started to make our mini bones, we could immediately see a difference. The mutant cells on the right-hand side make a very thin thread-like construct. They're very contractile. They pull the bone into a thread, and they then go on to form perfectly normal bones. But this tells us that there's some kind of mechanical sensing defect in these cells, which was unsuspected and which nobody knows about. And this may be druggable. So we're very excited if we could follow this up and understand it a bit better.
So they clearly look very different, I guess, and so I guess the extension is that they're modeling some aspect of bone formation that in Paget's disease. So do you have any idea of what bone, at this stage, what bone cells are affected? Would it be
Well,
the
what we do know is that it's not the osteoclasts. So it's not the major drug targets of the bisphosphonates. There are no osteoclasts in this model. So this is telling us something completely new. It's the bones at this stage contain both osteoblasts and osteocytes, but no osteoclasts. So this is a new avenue to discover mechanisms and drugs.
Yeah, that's really exciting, because I think there is work that suggests, although Paget's is an osteoclast disorder, there are these secondary involvements of osteoblasts and other cell types, and actually they might be some of the drivers of the Pagetic phenotype. So that's a really powerful model, and to see such a big difference is really impressive. So can you study the mechanical properties of these bones in the same way that Pagetic bone in people affected perhaps is more susceptible to fractures? Is there any way of looking at bone strength or bone integrity in this sort of model, do you think?
There is. I think at the moment our main interest is how the cells respond to mechanical load, because that may be driving the distortion. The bones are feeling the mechanics differently and responding by overgrowing and making bulges and lumps instead of a nice flat surface. So we want to study the mechanosensing and mechanotransduction pathways that help the cells to interpret their mechanical environment, and we load them with ultrasound. They do respond to ultrasound with standard load-bearing pathway characteristics. And of course, their physical properties will change over time. But again, to get a really nice mineralized bone, we probably need to run them out to 5 months. And that takes a bit of time and funding.
Indeed, it does. So, so I guess just to round off, then, I think you've touched on the kind of potential of this sort of model for studying Paget's, and also more broadly, perhaps studying other bone disorders. What do you see as maybe the sort of 5-year vision? What potentially might we be able to do as a field when we're able to establish these more physiologically relevant bone models?
It's very exciting in terms of drug discovery, because quite a number of rare bone diseases have well-known mutations of specific proteins. We can put these in very easily and flexibly into the models, and then start to understand in the context of a whole organ, a whole bone organ, rather than isolated cells, what the effects of those mutations are. And we will be able, using our fantastic sequencing resources in Oxford, to do single cell sequencing and atlas them so that we can actually map the specific cell types that occur throughout the lifetime of these little bones, and how they differ in disease and how they respond to therapy at a single cell level.
Well, thank you, Philippa, for that overview of your research. Really, really exciting. And I think there's some tremendous prospects for the future for Paget's disease and more broadly studying kind of the basic biology of other bone diseases, and as a platform potentially for therapeutic advances as well. So on behalf of the Paget's Association, we wish you every success with your continued research programme. And again, we thank you very much for joining us today for this presentation. Thank you.
Thank you.